Good afternoon, everyone. Myself, Zakaria Ahmed, Technical Assistant, Biotech Hub, Bahana College. I welcome you all here to our second day of the 3-day online, 3-day hands-on training program on bioinformatics, which is organized by Institutional Biotech Hub, Bahana College, Jorhat. And our today's resource person is Dr. Abhishek Das Gupta, sir. Now, I request Miss Malika Barua, Deputy Assistant, Bahana College, to introduce our resource person. Thank you to Miss Malika Barua.
Thank you, Zakaria. It gives me great pleasure to introduce Dr. Abhishek Das Gupta Sir, an accomplished biotechnologist and dedicated academician who currently serves as an assistant professor at the Center for Biotechnology and Bioinformatics, Dibrugarh University. Dr. Das Gupta's area of expertise lies in microbial ecology and bioinformatics, where his research explores how microorganisms adapt and thrive in hydrocarbon-contaminated environments. His work provides valuable insights into the natural processes of bioremediation, the use of microbes to clean up pollutants, which hold immense importance for environmental sustainability. In addition to his experimental research, Dr. Dasgupta effectively integrates computational tools and metagenomic approaches to better understand microbial diversity and ecological functions. Through his interdisciplinary approach, he bridges the gap between microbiology and data-driven bioinformatics, contributing significantly to both scientific understanding and environmental conservation. We are truly honored to have with us today Dr. Abhishek Dasgupta sir, whose contributions continue to inspire young researchers and advance the field of biotechnology and environmental microbiology. Please join me in extending a warm welcome to Dr. Abhishek Dasgupta.
Thank you so much, Miss Malika, for introducing our resource person. Now, I request our resource person, Dr. Abhishek Das Gupta, sir, to start the session. Thank you. Now, I like to hand over the session to you, sir. Sir, the session is
Thank
all
you.
yours, sir.
Yes. Thank you for the invitation. So, before starting the session, I just wanted to know how many participants are there.
Sir, they are joining now. There are 32. Sir,
32.
participants. Yes, sir.
So, I think if they have any questions during the training, they can post it in the comment box, right? Like if they have any queries.
So they can write in our chat box if they have any
Okay.
queries, or they can turn on their mic.
Yes. Thank you.
Or raise hand.
Yeah. So, thank you for this warm invitation. And now I'm starting my session. And as you all know, that I'm just this one disabling my camera because we have some internet connection. So if I share my slides, there will be some problems. So is it okay to disable my camera now?
Sir?
Is it okay to turn off my camera? Because if I share the slides
So
and the
it's okay.
camera.
So yes, it's okay.
Okay, thank you. Okay, so thank you. Now I'm starting my session, and my session is on metagenomics. So first of all, what we have to understand what is metagenomics.
নেটৱৰ্কটো বেয়া বুলি কৈছে
So I think you can see the screen, right?
নহয় মই শ্বেয়াৰ কৰিলে আকৌ মই ৰেকৰ্ডিং কৰি আছোঁ যে প্ৰব্লেম সেইটো
Hello.
য়েছ ছাৰ
Yes, sir. Yeah, the screen is accessible, right?
য়েছ ছাৰ
You can see my screen? Okay. So first of all, we have to understand what is metagenomics. So,
মই হেৰি কৰিব ন
before that,
ৰেকৰ্ডিং কৰি থাকোঁ
what we will learn today? We will learn
Build up to
this topic, that is what is metagenomics, advantages of metagenomics over traditional technique, types of metagenomics study, tips for performing Amplicon metagenomics, what are FAST-Q5, and after learning all this, we will do a short hands-on on time 2. So first of all, what is metagenomics? As we all know, metagenomics, meta means whole and genomics means study of genome. Metagenomics mainly focuses on the microbial communities. So what is, first of all, suppose let us consider this as an area. Let us consider this as an area, and we want to understand what are the microbes present. What are the microbes present in this area? So with the help of metagenomics, what we will do is that we will directly isolate the samples and we will extract the DNA and we will see what are the microbes present. So basically in metagenomics what happens, we isolate the DNA, isolate the samples, suppose, this is the slide. So what we do is that we take the sample, we isolate the DNA and then we sequence it and then we perform bioinformatics analysis. So this is the part of metagenomics study. And metagenomics has an advantage over traditional method. Now what is a traditional method? Traditional method is that the method that we are following from our ancient time. That is, if you want to understand a microbe, what we do is that we isolate the microbe, do a pure culture in the plate and then we study it. But in metagenomics, what happens is that we don't have to isolate the microbes in our synthetic media. What we can do is that we can directly understand the microbial diversity by sequencing it. That means the isolation of the microbe in the synthetic media is omitted in metagenomics study. So again we come to the definition, what is metagenomics? Meta means whole and genomics means study of the genome. And it mainly focuses on the microbial community. So basically this is the metagenomics. So metagenomics was first coined by Joe Handelsman in the year 2000, in the early 2000. Okay. So now we come to the types of metagenomics, advantages of metagenomics over traditional technique. Now, as I have already told you that traditional technique means that you are culturing the bacteria in synthetic media. But sometimes what happens that all bacteria are not culturable. That means it has been found that only 1% of bacteria can be cultured in the laboratory. But what happens to the rest? So it becomes unknown. Now, with the help of metagenomics, you can understand, you can know the diversity of 99.9% bacteria that are in a environmental samples. Okay. So, what is that? This is the main advantage of metagenomics over traditional method. Now, if there is an advantage, there may be also a disadvantage. So what are the basic cons, means like basic disadvantages of a metagenomics study? The first thing is that it requires a huge amount. The cost of sequencing, the cost of kits, they are little bit more. Next is that you have to know bioinformatics. Without understanding the command-based line, this thing becomes a problem. And the last and foremost is that you have to know basic computational tools, basic computational coding you have to understand. But nowadays, some online softwares have been developed that just little bit eases this problem. Okay. So this is the basic this one. Metagenomics and traditional, this is the basic differentiation between a metagenomics and a traditional method. Next we come to types of metagenomic study. Now what is the types of metagenomic study? There are two types. One is your Amplicon metagenomics and another is your shotgun metagenomics. What is Amplicon metagenomics? Amplicon metagenomics is that In Amplicon, what we do is that we sequence this marker gene, that is 16S RDNA. We sequence this marker gene. And based on the marker gene, we differentiate our samples. Basically, Amplicon metagenomics is overall the sequencing or amplification of those marker genes. Like for bacteria, cystinas, for fungi, ITS. So these things you amplify and you sequence it. But in your shotgun metagenomics, what you do is that you don't have to amplify it. You just directly take the sample, extract the DNA, and you give it to your machine. That is sequencing machine. That is the shotgun sequencing. Okay. Now, what happens is that when you use a shotgun sequencing, you can see the taxonomic plus functional profile. See here in my slide also you can see in taxonomic and functional profile. That means who are there in the sample and what function they can do. If your research question is like that, it's better you go for shotgun metagenome. That means what are the community, what is the microbial community and what functional profile they have. That is shotgun metagenomics, right? And Amplicon metagenomics, what happens is that you just go for the taxonomic profile. That means what all are present in your sample. That is the main difference. So first difference is that Amplicon and shotgun. Amplicon is that what are the taxonomy, means like what are the bacteria or what are the microbes present in your sample, you go for Amplicon metagenomics. And shotgun metagenomics means what are the microbes present in your sample plus what functions they are doing. That is shotgun metagenomics. Now, here also in shotgun metagenomics, the cost of shotgun metagenomics is little bit more than Amplicon metagenomics. And for us as a beginner, it's better to understand the Amplicon metagenomics first, and then we can go for shotgun metagenomics study. Okay. So now let's come to the workflow. Let's come to the workflow of shotgun metagenomics. Now we have understood what is metagenomics, advantages of metagenomics over traditional technique, types of metagenomics study, and then we will come to the workflow of the metagenomics sequence. See, here in this slide you will see that a person is collecting the sample. Sample may be anything. You can collect sample from soil, you can collect sample from your gut, you can collect sample from your river, water, anything. Or environmental sample you can collect, human sample you can collect, animal fishes sample you can collect, anything else. But your research goal should be to understand what are the bacterial community or microbial community present in your sample. Now, once you have collected the sample, what you do is that you go for sample pre-treatment. Now, why sample pre-treatment is necessary? Sample pre-treatment is basically necessary to to make your sample free from contaminants. Now, what may be the contaminants? Suppose in case of soil, humus may be present, or other contaminants, organic acids may be present, that may interfere with your PCR amplification. So for that reason, what we do is that we go for sample pre-treatment. For sample pre-treatment also there are various protocols available. You can find kits also, protocols also. Once you have pre-treated the sample, what you do is that you extract the DNA and you amplify the marker gene. Once you pre-treat the sample, what you do is that you extract the DNA and you amplify the marker gene. So what happens when we amplify the marker gene, and why it is necessary? Because as earlier I told you that Amplicon, has the name amplify, Amplicon. So you have to amplify the marker gene because you are identifying the microbes based on the marker gene. So for that reason what we are doing is that we are amplifying the marker gene. So for bacteria, this cystinase RDNA you are amplifying. Once you have amplified, what you do is that you do a sequencing analysis. Now, sequencing analysis, nowadays next-gen sequencers are present. Next-gen sequencers help you to analyze large amount of sample in a very less time. I will not go to what are sequencers because it will take, this is out of scope of this today's training. So I will just tell you that there are two types of sequencer. One is the Sanger sequencer, that's basic Sanger sequencer, and another is your Nexus sequencer. Nexus sequencer may be your Illumina MindSeq, HiSeq, Nanopore, and other SMRT. So these are the Nexus. So for metagenomic study, we do it in a next-gen sequencing. Okay. So now, once we have identified the marker gene, what we do is that we do a sequencing study. Now, this is where, as a bioinformatics, your part will come. After sequencing, you have to analyze the sample, and based on your analysis, you have to classify the microbe. So, as a bioinformatics, this is your part. Other parts are the work of a wet lab scientist. But as a dry lab scientist, this part will be your part. That is, you have to analyze the samples. Okay. So this is all about amplicon sequence. So now what we have done is that we have understand what is metagenomics. We have understand what is metagenomics. We have seen that what is the advantages of metagenomics over traditional technique. We have understand the types of metagenomics, and we have seen the steps for performing a metagenomic study. Okay. Now, now I have a question for the participants. So, can anyone tell me why, if you want to understand only the composition, microbial composition of a environmental sample, suppose of a lake or of a hydrocarbon of a refinery, what type of metagenomic study would you imply? Participants, can anyone tell me? Suppose you want to only understand the microbial composition, you don't want to understand the function of the microbe. What study would you imply on that sample? Does anyone? Am I audible?
Yes, sir.
Okay. So I have a question for the participants that suppose if you want to isolate, you want to understand the microbial composition. Yeah, amplicon. Very good. Dr. P. Ramachandran. Okay. Amplicon sequence. Yeah. So why we want to go for amplicon sequencing? Because that we only want to understand the microbial composition of the sample. But if we want to understand the functions of the sample, we will go for shotgun sequence. So this is clear, I think. Yeah. So next, what we do is that we have understood that what are the steps for performing the amplicon metagenomics. So again, I am repeating this. First, what we have to do, we have to collect the sample, then we have to pre-treat the sample, we have to amplify the marker gene, and then we have to go for the sequencing part. Now, as a bioinformatics, after sequencing, your parts come. Okay, so for the analysis part, people will require you if you want to do the analysis. So, now, before jumping on the hands-on, what we have to do is that we have to understand something. We have to understand. We have to understand what is FASTA, and what is fast. So we have to understand before and then starting our analysis, we have to understand what is FASTA and what is FASTQ. I hope everyone knows about the FASTA format. So FASTA format is a normal format that we do for our analysis when we do a blast or when we do a fragment design or testing. We use FASTA format. But when we are starting with our metagenomics analysis, what we do is that we use a FASTQ format. That is a FASTA file having a quality file. we do a fast queue, we have a fast queue format. These formats are generated by next gen sequencer. Okay, this format are generated by next gen sequencers. What they do is that this format is called, it is a faster format and a quality format. So as a combination, we call this as a fast queue format. Okay, so yeah. So this is the example of fast queue format. See, this line, these are the, how many lines are there? 4 lines. 1, 2, 3 and 4. So this is an example of a fast queue format. So when you are working with a sequencer, that is next gen sequencer, you will find this format. Okay. So the first line, this is the first line. The first line is a called the, this is an identifier line. Identifier means the sequencer name, your sample ID, these things are all written in the first line. Now, your main sequence is in your second line. See, here you will see A, G, 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 A, G, 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 A, G, G, G. So this is a main sequence. Okay. So this is separator line and this one is your thread quality. This is a quality score. Based on this quality score, you have to analyze your sample. This quality score is very important. Now, what is this quality score? Quality score means that in sequencing, sometimes we have low quality sample. That means your contaminants. There are contaminants you are in your sequence. So this quality score helps you to identify the sequences that are very good and sequences that have contaminants. Now, what type of contaminants can come? See, when we are doing a next-gen sequencing, we have some barcodes, we have some linkages, this thing. Just remember that these things are required for you are adding some chemicals. These chemicals are required for your sequencing purpose. So what happens that these chemicals, when these chemicals are not removed from your amplified sequence, so they remain attached to your sequence and they contaminate. That means your sequence is not up to the mark. So these are the contaminants. So after sequencing also there is a software that checks whether your sequence is good or not and what score it will give. Okay. So based on the score, if your score that is Q32 is if your quality score is above 30, we consider it as a good sequence. Okay. If your quality score is above 40, we consider it as an excellent sequence. And if your quality score is very poor, that is Q2, very poor, we discard the sequence. So here also you will see this is called an ASCII code, ASCII code. Okay. So see here here you will see A. That means this sequence is 32. This is a good quality. That is G. This is a good quality sequence. And then we come to H. That is excellent quality. This up to this mark. That is TH, AH, TH, GH. These are what? These are the excellent quality sequence. And then I come to I. Can you okay? So there is no I. Anyways, so this is the main thing of a FASTQ file. So FASTQ file starts with an identifier. That means sequence identifier. That means it has a flow sale, sequence number, and your sample ID number. This is the identifier. Next, you have the sequence. Your sequence. This lane is a sequence lane. Okay. And next, you have a separator. See the plus sign. This is called the separator. And after that, you have the thread quality score. After that, you have the quality score. Okay. So these are the 4 things of a FASTQ file. Now, we have understood what is a FASTQ file. Now, my question is to you is that, now, can anyone tell me, suppose if my quality score is above 50, should I consider this FASTQ file as a good or excellent? Yeah, I have a question, I think. Anisha. Yeah, Anisha. I think someone has registered that. Anisha, you have a question, right? Hello. Yeah, Anisha has raised her hand. Has raised her hand. She has a question, I think.
So I think it was by mistake.
Okay. So, where was I? Yeah. So, if my quality score is above 50, so what should I consider? Should I consider it as a good or excellent? Yeah, somebody has told excellent. Yeah, we should consider it as an excellent. Yeah, Devashmita, yeah. We should consider it as an excellent. Now, if my quality score is low, like 25, should I consider it or should I discard it? Like it is between 25 to 30. Should I consider it or should I discard it? Discard this. What do we do in that case? Suppose you have a quality, yeah, we should discard it. Yeah, discard it. Good. So what we will do is that we should always keep in mind that the more quality we have, the more good will be our analysis. Otherwise, we have a bias. Okay. So the more quality we have, the more good will be our analysis. Otherwise, we will have a bias in the sample. Okay. So this is all about the theoretical part. This is all about the theoretical part. What is class-Q? What is metagenomics? What is amplicon sequencing? What is shotgun sequencing? This is all about the
Okay, sir.
theoretical part. Now next we move on to our hands-on. So uh, what we will do is that I will just give you a walkthrough. Now, first of all, there is a library called KIND. Okay, what is a KIND? So this is a type. Time is something that once you get the sequence, it helps you in the analysis of the sequence. Basically, this time, this software is an open source. You don't have to pay a single penny there. Okay, so this is an open source. So that's why it is popular among the researchers. First. Second thing is that this kind is used for amplicon sequencing. Okay, now this software has also developed itself in the recent times. When I did my PhD in Kind, that was a different thing, and now Kind has developed. It has integrated AI and everything. Okay, so now we will do this moving pictures tutorial from Kind. So for that, first of all, what we do is we will have to have a Linux. Okay, the Kind only runs in Linux. Windows, if all of you, I guess all of you have Windows system or you have Linux. You can comment in the comment box if you have Windows or if you have Linux. Okay. So if you have Windows or if you have Linux, it's well and good. If you have Windows, what we have to do is that first of all, what we have to do is that we have to go to the control panel. Okay, control panel. First of all, we have to go to the control panel and we have to open it. Open the control panel. Now, next what we have to do is that we have to go to the turn Windows feature on and off. We have to go there, turn Windows feature on and off. Then we have to do is that we have to search for WSL, Windows Subsystem Linux. See here, this feature is enabled in my here. This is Windows Subsystem Linux. You have to switch on Windows Subsystem Linux. Okay, just click it here and close it. Okay, and then you have to reboot your system, right? And then yeah. So then what we have to do is that we have to go to the this one Microsoft Store, and then you have to download the Ubuntu. This is the initial part. Suppose I assume you have the Ubuntu and I will show you how we have to understand, how we have to do the Linux part. Okay, how we have to install the Linux part. So once we have installed the Linux, so we will see the terminal like this. This is the terminal. In Linux, this is called the terminal, where we do the command part. This in Linux, this is called the terminal where we do the command part. Okay. Now, what we will do is that we will start with this tutorial, that is moving pictures tutorial. So what we will learn in this tutorial? In this tutorial, we will use what we will do is that we will understand how Amplicon sequencing is done. How Amplicon sequencing is done, we will understand in this tutorial. So for that, what we have to do, we have to, first of all, we have to activate, we have to open our Kine. So what we do is that, how we open our Kine, we just write here, Kanda activate. This is the first command of activating Kine. Already I have activated my Kine, that is my Kine is running. Just we type the command Kanda activate Kine. Once you have installed everything, what you need for running Kine, you need this one. For running Kine, you need a Windows Subsystem Linux. I am telling about Windows. You need to require a Windows Subsystem Linux, and you require a Windows Subsystem Linux, and you require Ubuntu. And once you have installed everything, you have Windows WSL, and you have installed everything Ubuntu, then what you do is that you install Kine. So after finishing our hands-on, after this session, what I will do is that I will share with you the document, how to install everything in your system. Now what you do is that you just see how we are installing and how we are running the code. Okay. So you have installed everything. You have an Ubuntu system. You have installed Linux, and you have a Kine 2 in your system. Now what we do is that we have to open Kine 2. This is the terminal. I have opened the terminal. Now what I will do is that I have to activate. That means this command I am typing, Conda activate Kine 2. That means I am opening my Kine 2 software. Once I type this command, the Kine 2 started running. See here you will find a base written. That means your Kine 2 is not activated. Once you activate it, here it comes Kine 2. Now, next what we have to do, we have to go to our directory. Here I have made a directory for the tutorial. My directory name is Q tutorial. Okay, my directory name is Q tutorial. So what I am doing is that I am changing my directory. This is called a directory. All of those who are new, I guess most of you are new, in Linux directory is same as a folder. What I am doing is that I am changing my folder. Currently I am in the E folder, and I am changing my folder to the Q tutorial folder. This Q tutorial folder I have made for the training purpose. See, this is the Q tutorial folder that I have made for the training purposes. Okay, so this is the time to installation thing. So first what you will do is that you will open the start menu, search turn Windows feature on and off, and as I have shown in the list feature, you have to scroll down and check box to the next to the Windows subsystem for Linux, and then you have to click OK and restart your computer. So what we require, we have Windows subsystem for Linux. This thing I am telling about the Windows only. Okay. Now next part we have to install Ubuntu. What is Ubuntu? Ubuntu operates on Linux operating system. So Kine 2 basically runs on Linux operating system. So what we do is that next thing what we do, we install Ubuntu. How we will install Ubuntu? We will go to the Microsoft Store, click on Ubuntu and click Get. Launch it, and once it gets downloaded, again we have to reboot the system. Next what we do is that we install the Miniconda. So this thing we just have to install it. Okay, we have to install the Miniconda. These things are required for running your Kine. You have to have WSL, you have to have Ubuntu, and you have to install the Miniconda version. Now, once we have installed everything, next what we do is that we start our timestamp. Okay. So now, where was I? Yeah. So Q tutorial. So this Q tutorial I have made for the tutorial purpose. I am in the E drive. I am in the E drive. See here, E drive. And I have to go to the Q tutorial. So what I will do, I will just change the directory. That means I will just write CD Q tutorial and I will hit here enter. And now I am in the Q tutorial file directory. Now I have to see what are the files present in the directory. Just I will write here ls and I will just press enter. See, so these are the files. That is, these are the files. Time installation guide, document files. So these are the files present. Now when you check Q tutorial, you will see everything same, similar. Okay, that's a similar thing, right? So now this is all about how we have to change the directory. Now, again, what I want to do is that I want to make a new directory for your this hands-on. What I will do, I will just write here mkdir, and what I will do is that I will just make a new directory. Suppose the directory what I have given here. I will just make a new directory and I will just write here EMP. Okay, I will just write here EMP. And now I will just write here ls. See, this is a directory EMP. These are not bold, that means these are the files. Again, in the Ubuntu terminal, what you will see that the things that are bold, these are called a directory, and the things that are not bold, these are the files. Similar, that is, remember this directory is a file, folder, and these are the files. Okay. So, first of all, what we do is that we write here mkdir, that is make directory emp, and then we will see ls. Now I want to go to this folder. EMP, what I mean, I will just write here cd emp. Now I am inside this folder emp ls. If I type, I will not get it. So here, so see, I am inside this folder. That is, I am inside the emp folder. Okay. So this is the thing. Now we have to understand. We have to start our analysis part. This was all about making the directory, changing the directory, installing Ubuntu, installing Linux, everything. These are the. Now we have to understand. We will start with the analysis part. So before starting with the analysis part, what we have to do is that we have to understand what are the things we will require. The first and foremost, we have to, when we collect the sample, we also collect the sample metadata. When we collect the sample, we also collect the sample metadata. So, in this part, see, this is the sample metadata. I am just showing you. So, see, this is how a sample metadata has been collected. So, this is the name of the sample. What is this? These are the names of the sample. This is the name of the sample. L158, L1557, L1576, L1505, L1, L2155, L2175, L25204, L5222, L5322. So these are what? These are the sample IDs. These are what? These are the barcode sequences. Okay, just remember every sequence has its own barcode. That means in sequencer also we have some barcodes. Barcodes are attached to sequencers so that they can know that from which sequence belongs to which sample. So what they do is that they attach their own barcode sequence. Okay, and this is the body side from which you have taken the sample. So actually this sample is a human sample. Okay. So this is the body side they have taken, that is from the gut they have taken, palm they have taken, tongue they have taken. So this is how. So when you are analyzing a metagenomic sample, what you have to do is that you have to isolate the sample. But with that you have to collect the metadata. That means you have to have the metadata. That means from where the sample has been collected, which is the time, which is the type, everything. So here it is about the human sample. But suppose, so this is the year, this is the month, day, subject one, which is the subject, that means this is experiment. So these are the things you have to do. Okay. So now antibiotics, whether these subjects were treated with antibiotics, yes or no, days is experiment. So now what we have to do, suppose we are collecting a soil sample. So soil sample, what we will do, we will see that what is the name of the geographical location, latitude, longitude. Then we have to collect the soil type, from which part you have collected, whether you have collected a bulk sample or you have collected a rhizospheric sample, or what are the is there any heavy metal. So everything. So this everything combines with the sample method. So while collecting sample, you have to keep in mind two things. That means you have to collect the sample, and with that you have to also collect the sample metadata. Okay, so you have to collect a notebook, and the notebook you have to write everything about the metadata part, because this sample metadata will be required when you do the analysis part. Okay, so this is all about the sample method. Now, this is the code of sample. So basically, the sample we are analyzing is that this is a sample of a human being or a subject that have been treated with antibiotic and different. That means the persons that have been treated with antibiotic and the persons that are not treated with antibiotic. So what we are doing is that we are analyzing their microbes. See, subject 1 has been treated with antibiotic. Okay, this subject has not been treated with antibiotic. Some of them have treated with antibiotic. So what we've done, we have collected their samples and we have isolated the microbes. That is, amphibian metagenomes we have isolated. We have done the amphibian metagenomes part. Now, as a bioinformatician, what our work is to do, we have to analyze the sample. That means we have to see whether there is any difference between the samples or whether we have any what is the taxonomy completion. So these things we have to sequence, like this will be our part. This will be our analysis part. So now when we start our analysis, what we have to do is that we have to download the sample metadata. Okay. So how we will just a minute. Okay. So how we will analyze means how we will download the sample metadata. This is the command wget. So wget is a Linux command where we can download from the internet, directly from the internet. So in Windows what we just go here and we click download, right? So in Linux we just write it here wget and it will already be downloaded. So now I have clicked it here, I have copied it, and let's see. I have clicked it here and it's downloaded. Okay. So where it is downloaded? It is downloaded in your EMP folder. Now let us see. This is the EMP folder. Yeah, it has been downloaded. Okay. This is the way of checking. Otherwise, what you can do is that you can type there ls and you can see your sample metadata has been downloaded in the EMP folder. Right? So this is the first part. You have downloaded the sample metadata. Now, next part is what? Next part is you have to convert it into a KIME format. See, this is QZB. What is this for? Now, when we are working with a text file, we have some file extension, right? When we work with a text file, what we save it as? We save it as .txt, right? .txt. When we are working with a FASTA file, we write here as .fasta. Or when we are working with a FASTQ file, we write here .fastq. So KIME is what? KIME format is .qzp. qzp means when we have downloaded the output file, you want to check the output file. How do we check? Where you will check? We check in the KIME here in the KIME web page. So when you are checking this thing in the KIME web browser, KIME web browser can only recognize QZV and QZA format. These two format only recognizes. So what we do is that we convert our file into QZV and QZA format. Okay. Just remember, KIME can recognize only two formats, that is QZV and another format is QZA. So what we do is that we only do this. We when we have our sample and we want to check it in the Kind software, Kind web browser, what we do is that we change our format to QZV or QZA. Okay. So what we do, we just click it here. I just copy this and I run it here and I press enter. Okay, so while this thing learn, what I will do is that I will just open another thing. This thing is required for see time to view. Here you can view your time file directly. Okay. So what I view is view.kind2.org, where you can directly view your files, right? So these are some of the examples they have given: alpha diversity, exploring taxonomy of the files, ordination plot. So these are some of the things they have given. So now let's see our file is ready or not. Yeah, just a minute. So just do one thing. I will just move to here. Yeah, so this is how a sample QZB interactive thing looks. See, these are the what? I have converted my file to QZB and just drag it and drop it here. So this is what? This is the sample ID, right? And these are the barcode sequences. And these are the sites, the left palm, right palm. These are the sites where we have isolated the bacteria. Which year we have isolated in 2009, which is the month, which is the day. Everything we have to document. Means documentation is also a part of your metagenomic study. Not only analysis, for analyzing also you have to document everything. Okay. Now, so this is how your sample metadata looks in the Kind view. So remember, once you have downloaded Kind, you can also check your files with view.kind2.org. Okay. Now, next what we do is that, next we have this one. We have converted our sample metadata. Now, once we have converted our sample metadata, what we will do? We will have to require our sample, right? So, here I am working with an EMP sample. So, there are different types of samples. When you are working with NextGen, there are paired sequence, single sequence, like EMP, Cassava. These are the different types of formats. So, this is these things we have to like when you are doing a metagenomic study, you have also to learn all these things. That is, what is the sequencer, what is the NextGen sequencer, what are the formats generated, sequence formats generated by the sequencer. These things you have to understand. Okay. So now what I am doing is that I am now, since see, I have already told you what is wget. Wget means you can directly download the sequence from the internet. So without clicking this thing, what we do is that we just write here wget. So once I have downloaded my sample metadata, next what I am going to do is that I want to download my sample. So EMP sample. So what I will do, I will just copy my code here. I will just copy my code here and I will just click it here and see it's downloaded. It will take some time, let it download. So it's downloaded. So since we are doing, I am just doing the Kind tutorial I am downloading. This thing you just see, you don't have to, when you are working with your own sequence, you don't have to download. It will already be available with you in your laptop on your desktop. Okay. So now this part is important. That means, see, FASTQ sequence have a huge. They have using. They have. I think they utilize a lot of memory. So what companies or what sequencing companies do is that when they provide you with the sequence, they provide with a zip thing, like a zip folder. Okay, so that means they compress it and they provide it. Okay, but when you are working in time, you have to unzip it. Up to this, it was already all downloading the sequences. Now, this thing, unzip DEMP single end sequence, EMP single end sequence.zip. That means what I am telling the computer is that you unzip the sequence. That means unzip EMP single end sequence.zip. Okay. My sequence name is EMP single end sequence. And you unzip it and you compress, means like you unzip it. You just decompress. Okay, and now I'm clicking in. Now everything has been unzipped, right? See, in MEMP single and sequence or fast queue, these are everything has been unzipped. Barcode annual sequence, everything has been unzipped. It has been decompressed. Now let me check. Yes. So these are unzipped. Okay. These are the barcode FASTQ file, and this is the sequence.FASTQ file. See, 24,711 KB and this is 3,000 INC. So this is the file size. And when you don't unzip totally, it will look like this. Okay. So once you have done this thing, we have unzipped it. Now our next step will be is that we have to import it. That means till now, what we have done, our data is not on time. What we were doing, we just unzipping our data. But to perform a kind analysis or to perform an analysis, we have to import it. Similar thing, just remember, importing means what? Suppose you are working with Word file, right? What we do is that, suppose this is the Word file. What we do is that we just go here, and we just import it, right? We just import it. So this is the Word file. So this is the kind of importing. Okay. So now what we have to do, we have to import the file in our time software. So how we will import it? We will use this command, C, time to import. This command is required to import your sequence into time format. Earlier what I told, there are two formats, that is QZA and QZB. QZB is used for visualization. QZB, that means so QZV. This V part, this is for visualization purposes. So kind has two formats, that is QZA and QZV. So QZV is used for visualization purposes. Okay. Now we have to import our sequence. We have unzipped our sequence and now we have to import our sequence in our kind software. What we will do is that we will use the kind to import. Just I am copying this code and I am pasting it. And I am pasting. Output will be EMP single and QZA. That will be our output. Okay. So our file has been imported. Now, what will be our next step? Next step is demultiplexing. This is the important step. What is demultiplexing? Demultiplexing means we have the barcode sequence. What we have seen is that we have the barcode sequence. So these are the barcode sequence. These sequences are not necessary. These are the instrument sequence that just they have put the barcode, and these sequences are not necessary. This step, this demultiplexing means that you have to cut the barcode sequence from your EN sequence. Suppose this is the barcode sequence and this is the EN sequence. So after demultiplexing, what happens is that your main aim is to just get the DNA sequence for the analysis. You don't require this sequence. So what you have to do, you have to cut it. That is demultiplex. That term is called a demultiplex. So when you are working with metagenomics, you will find some terms, that is demultiplexing, deionizing, deep blood, this one, that too. So these are what feature table, OQ table. So these are some of the terms that you have to be familiar with yourself when you are working with a metagenomic sample. Okay. So now what we do is that we come to the demultiplexing part. So now demultiplexing happens. What is the main thing? For demultiplexing, our main thing, we have to cut this sample. That means we don't require this barcode sequence. We have to cut the barcode sequence and we just require this DNA sequence. Okay. Now, you just require the DNA sequence. So what for demultiplexing, this is the command. Time demux EMP single. So if it was paired end, we would have write the time demux EMP paired. Or if it was cassava, we would write time demux EMP cassava. What is I here? This is the input file. See, what is I here? This is the input file. What you are inputting QZ file, EMP single end sequence.qza. This was the file that you got as an output, EMP single end sequence.qza. So what you are doing is that you are inputting this QZ file and your barcode file, you are telling the computer take the barcode file from the sample metadata TSV. That sample metadata TSV that you have downloaded, this barcode file, you are telling the computer to take the barcode file from there. And you are also telling the computer that the barcode column will be your barcode sequence. Everything you have to specify. See, when you are writing your code, you have to specify everything. It is similar thing. Some of us are afraid of writing code, but writing code is also similar. That is, you are telling your computer does not know or anything about it. Just you have to do is that you have to tell it that you have to do it, you have to do like this, like this. So this thing you have to write in a code. All you have to know is the syntax of the code, whether your code writing is good or means like you are writing the proper syntax or not. Every programming language has its syntax. Syntax means rules and for writing the code. So here also, whether you are writing the code, it is the main. You have to understand syntax of the code. And so now what output name is sample sequence demux.qza. That will be your output sequence. And if there is any error, it will be demux.qza. That means why there is error or not. So basically, see what we have done. We have written the command demux emp single. That is the command. We have an input sequence. Input sequence means this one. We have what we are inputting. That means we are inputting EMP single and sequence of qza. We have a barcode file, sample metadata.tsv. We have a barcode column, barcode sequence. We have a sample sequence and everything. So what you require, you require an input file, you require a barcode file. That means what will be the barcode. You have to tell the computer that which column you are considering as a barcode, barcode.sequence, and you have to tell the output, what output name you require. Here my output name I require as sequence demux.dza, and if there is an error where the error file details will be stored, it will store in demux.detail.dza. Okay, now let us run this code and we will see the what happens. Now the code is run. It will take some time. Now, after demuxing, that means after you have this one, you have demuxed the barcode sample. We have to what we have to do? We have to just visualize. Okay, QZ file you cannot visualize it, you will just convert it into a QCD file. Now after demuxing, we have to start with the feature table. Now feature table is a very important. Feature table means, suppose you have isolated the sample and which feature? Feature means the microbes. We are giving here microbes some numbers, that is ID, that is 1, 2, 3, 4, 5, 6. Which ID belongs to which sequence? So this is the feature table. Okay. Let me show you. Anyways, see, your demux file has been generated, right? Now, what our next step is that we have to convert it into a QZB file, where we can see this one, where we can see our analysis part, what is happening, or where we can visualize everything. So, what we will do is that we will just summarize it. See, kind demux summarize. That means it is summarizing your result. Okay. So how we will summarize as a table? What you will do? Input data. It is taking the input data from demux.qza. From here it is taking the input data, and the visualization part is demux.qza. So when we want to visualize it, we will convert it into a QZB file. Let's do it.
Serialism.
Yeah. So this one we have got the now. Next part is that if you want to see, you can also see it, what the results are all about. Was it? Yeah. So see, this is the demultiplex sequence count. That means what are the sequences that have been demultiplexed. That means what are barcodes have been removed. So total sequence you got this 263931. This is a very good. The more sequence you get, the more quality, means like your analysis quality will be good. That is 263931. This is the total sequence. So this all gives you the quality report. What is the minimum of reads? What is the median of reads? What is the maximum of reads? So this is the, always just try to look at this. Okay. So now you need the total number of reads you have 263 931. That means it's a load. You have 32 samples and you have this much of it. That means most of the samples have been covered. Okay. So here you can check that total 34 sample, which sample has the highest number of reads that has crossed the limit. So So LS4S112, this has the highest number. That means that PVT, it is a good sample. It has a good number of the barcode sequence were. Okay. So now there is an interactive plot also. You can see the plot was generated. There is a quality score. See, earlier I told you about the quality score. That is thread quality 30, thread quality 40, thread quality 10. So this is the thing. This is a sequence bit. That is the number of sequence, and this is the quality score. This sequence, earlier sequence, these are the good quality score. This quality. So up to, you can consider like this. Suppose this is the 25 quality score, right? And you want to consider it 30. You will see. So up to 100, up to 110 the spare you can consider. Okay, this thing they have the low quality, so you can discard it. You can discard. Up to this you can consider, and less you can discard. Okay. So this is how we check the quality score. Okay, so these are all of the things, that is whisker plot, bottom of the, these are some of the things. So for this thing, you just have to see the graph. Okay, you have to check that up to how many samples sequence base you can take. So I think up to this. If you consider this 30, you can take up to this. If you consider less, you can take up to this. Okay. So this is all about checking the quality score and demands. Now, once we have done the demands, next what we will do is that we will check the sequence quality and feature table construction. That means earlier in the demands we have seen the quality score. Now based on the quality score, what we will do, we will take only those samples that have good quality and those reads that don't have the, we will just discard them. So this is the step. That is the next step, is that sequence quality control and feature table construction. That means you are discarding the samples that have the reads that have low quality score, and you are taking the reads only that have the highest quality score. And after that what you are doing, you are constructing a feature table. That means you are constructing a table that will be used in your taxonomic classification. Now, what is a feature table? Feature table means, suppose, just I'm giving an example. Suppose there are 3 bacteria, 1, 2, 3, and you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 reads. Okay. This 3 thing, when you consider a feature table, that is, these 3 reads have a feature that is equivalent to 1. This thing has a feature that is equivalent to 2. And this thing has a feature that is equivalent to 3. So this is a feature table. Okay. Feature means what? Feature means the bacteria that has the feature. So this is a unique table. This is the starting point of your taxonomic classification. First, what you have to do, you have to construct a feature table, and based on the feature table, you have to do the taxonomic classification part. Okay. So this is the thing. Suppose 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. These are the reads you have, and you are constructing a feature table. So according to the property, these reads will be assigned the feature ID number, and this ID number is unique. It is not like all the bacteria, some of the bacteria will have its no. These are unique to the bacteria. Okay, bacterial species. So this will be the unique ID. Okay. Now we have to consider, next we have to do what? We have to do the sequence quality control, and we have to start with the feature table. Okay. For that, what we will use? We will use a pipeline. Pipeline is what? In computational terms, pipelines means step by step. Suppose, see, these are the steps, right? These are the steps we have to do. Suppose what we do is that when we are for this workshop, when we are logging in, what we do? We open our Gmail account, we click the link, then we type in the link, and then we log in, right? So what is this? We are doing step by step. That we have to do a single step at a single time. What will happen if we do all the things and we just write a command and everything happens? You don't have to do each step at a single time. Everything happens at a flow. So this is called a pipeline. That means in a pipeline you just write a single command and everything will happen in a flow. So that too is a pipeline that is for detecting and correcting chimera, amplitude sequence. That means for chimera. Now what is a chimera? Chimera, as I earlier told you, that means a sequence, you have a DNA sequence and your DNA sequence has been attached to a different artifact. That means those things that are not required for your analysis thing. It may be a PCA, it may be a tag polymerase sequence. It has been, sometimes it happens. Okay. So these are what? These are the chimeras. So what we do is that DNA is for detecting and connecting where possible amplitude sequence data. So this is the pipeline. So we consider this as a pipeline. So what is a pipeline? Pipeline means when you write a command, everything happens. Each step it occurs automatically. You don't have to write single command and single step. I will just write a single command and each step. That means it will detect. It will also detect whether there is a chimeric sequence or not, and also it will correct the sequence. Everything will happen. Okay. So now see, this is the command. Time that to DNA single, because our sequence is a single read, it is not a pair read, but a single sequence we are working on. Pair read means when we have two sequences, like just a minute. Suppose this is a sequence, we call it as a forward sequence, and this will be your reverse sequence. So this is a pair read. Okay, this is a forward sequence and this is a reverse sequence. So single sequence we are working. So what we will do? We just write here a single sequence. So what we are writing here? We are writing here that to DNA single. Next, our inputs. What is our input? Here our input is demultiplex sequence demultiplex. So the things what we got from demultiplexing, we will use this as an input. So time also goes step by step. That means first what you will require, you will require your demultiplexer. You cannot jump into the that group without demultiplexing your sequence. So demultiplexing your sequence, and now you are writing here P trim length 0 and P trunk length 120. That means what? See, just a minute. Yeah. So 120, that means up to this we are cutting the sequence. Okay, up to this they have cut the sequence, but we can cut it also up to this 30. But still we are doing, because here we have what we require a little bit this one, because you have sequencing V3 and V4 regions. So for that you will their length is little bit longer. So now see, what we have written here is that trim length 0. That means from here you don't require, it is 0. It will start from here. Okay, and then up to length trunk. Where we have to cut up to 120. Where we have to cut up to 120. Okay, so now computer what we do, time what we will do, it will cut the sequence and will remove the sequence. Okay. Now, representative sequence or exact sequence is already. So, representative sequence means what? Those sequences that have been cut, it will be this one in QZA. Okay, it will be outputted in QZ. Table. The table will be outputted in table.qza, and the D-argonic stats. Those stats that we will require, like this stats. Like this stats, we require to understand. We will require the stats that what sequence was present, what sequence has been cut. This will be done in That use of A. So there will be three outputs. That is a sequence. The sequence that you will require for your further downstream, that will be outputted in RepSec QZA. The table will be in feature table will be in QZA and table.qza, and you will get a linearizing stat. That means you will get a sequence as well as the feature table. Okay. So let's run it. Take some time. So once we done this, what we will do is that we will convert our input file. That means the stats we will require, because we have to check that what are the sequence they have been cut, what everything we have to check the stats. So we will convert the stats into a visualization file. So now after converting this, after doing this, we will go to the feature table and feature data. That means what we will do from there, the table that we will got and the sample metadata we will what we will convert a feature table. That means after filtering, after removing the unwanted sequence, unwanted reads, we will take our reads and we will we will draw the feature table and we will summarize the table. Okay. And we will summarize the table. And after summarizing, what we will do is that we will directly go to the taxonomic analysis. After summarizing, we will directly go to the taxonomic analysis. Taxonomic analysis means what are the bacteria that are present. So based on a feature table only, you can analyze the taxonomy. You cannot directly go to the taxonomy. You have to make the feature table and then only you have to go to the taxonomy part. Okay. And after doing the taxonomy part, you will check that what are the what are the bacteria that were present in the gut. You will just see in the subject 1 and subject 2 and you will check that subject 1 what are the bacteria dominantly present, and you will get the subject what are the bacteria that are not dominantly present. Okay, so now we got the stats of QC. Next what we will do is that yeah. So now we will convert this stats.qza to stats.qz and we will so now see our visualization stats.qz has been saved. Let us go there and yeah. So see, first sample, this is a sample ID L1S105. This is the input reads that were currently inputted. Okay, that you had earlier. These were the filtered reads. 8571 they were filtered. So after the first primary filtration, this percentage of the reads have passed. That is 75.58 have been passed. Now this percentage of the read 8499 have been cut. That means they were not like they contain contaminants. So they have been removed. Lastly, you get this, that is 7780. That is the percentage is 68.61. So see, from 75.58 your percentage has been 68.61. What you can do is that you can calculate what is the percentage of the this one. What will be the total? Means what is the read percentage? That means what is the total percentage? When total input you had and what is the final percentage? You can calculate, and lastly you can calculate the percentage, how much of your percentage of reads have been passed the filter. Based on that, see, most of the things have been passed. We have a good the sequence in that is a good quality. 68, 73, 71, 77, 74. These are the good quality. Now this one, 38. That means most of the have been not passed. That is LS22. So this is little bit less quality. Means like this will present a bias. Bias, what I am telling is that if your reads percentage of the sample of the reads of percentage is 80, 90, that means most of the sample, most of the bacteria have been covered from the sample. Now, if your read percentage becomes less, that means 40, 30, that means only few number, few areas, like few bacterial samples will be covered, bacterial community will be covered. So that means there will be a bias. You cannot tell from the sample that this bacterial community. So these are the communities in your sample. So more percentage of reads, more good will be your analysis. This is your basic thing. Okay. So now what we have done is that we have this one. This is the stat usability part. Now next what we will do, we will go to the feature table. As I already told you, feature table means we are based on the quality, on the reads, your time will what? Time will annotate. That means it will annotate your sample to the feature ID. Till now, we don't know about the bacteria. We just, what we will do, we will just annotate the sample to the feature ID. That means this sample, this read belongs to ID1, this read belongs to ID2, this read belongs to ID3, this read belongs to ID2, 4. So that means what they are annotating is your reads to the IDs. Okay. So similar here, what we will require? We will require an input file. Input file, what you are doing, you are taking the input file to the table.usa. Table.usa, where you got T here. After that two, that is table. So here you are taking that table.usa. Okay. And data sample and your time feature table, this one, visualization, table.usa. Similarly, your feature table, you are, what you are doing, revsec, that data you got, and you are visualizing it, revsec.usa. Okay. So now what we have done, we got our table.usa. Next, what we will do, we will get our revsec.usa. Now see, there are two commands we are typing, right? One is feature table summarize and feature table tabulate. See, feature table summarize will give you information on how many sequences are associated with each sample, each feature. That means suppose my sample I have 10 sequence, 10 features. That means bacterial sequence or 15 bacterial sequence are associated with my first sample, and then in a second sample I have 20 bacterial sequence associated. In my third sample, I have 30 bacterial sequences associated. So this command, what it summarizes and it shows us that how many sequences are associated with each sample. And feature table tablet, what it does, it will provide now feature ID. It will do a blast search and it will provide that which feature ID belongs to each sample. So there is a difference. Feature table summarize what it does. It just tells you what sequences are associated with your sample, and feature table tabulate, it does a blast search. See here it is written. It does a blast search and it will provide mapping of the feature ID to the sequences. Okay. Let's see. Yeah. So you go through that. That is table.qzb and you get the website.qz. First, what we will do is that first we will check the table.qzb. Now, see. So, what is the frequency? Frequency of each per sample. That is minimum frequency is 897 first quarter. So these are the statistical things. Frequency per sample. Let's check this. See, these are the sequences that are associated with the number of samples. Here are the sequences named. That is A, B, C, D. These are the sequences. What is the frequency of each sequence associated with this sample? That is 9820. This second sample 9740. Now this is the frequency. You can also see the feature details. These are some of the frequencies. These are the feature details. These are the features. Okay. You don't know. Still you don't know that which feature belongs to which feature ID. They are only the feature. That means these are the properties. Just assume these are the properties. But you don't know which property belongs to which person. Now in our next table we will see that what are the properties. What are the person's name. Okay. So these are the frequency. This is the frequency. Our sample observed out of 34. This thing, this feature is observed in having a frequency of 11,373 and is observed in how many samples? 13 samples. This feature, this is also a feature. This feature has a frequency of 8,929 and is observed in 16 samples. This is also a feature. This feature has a frequency of 8,806 and is observed in 25 samples. So this is the frequency and this is the number of samples observed. Okay. Now let's move on to the website. Okay, let's move on to the website part. Let's see website part user. So now see, this is the feature ID that we got and this is the sequence name, right? This is the sequence that has been blasted and this is the length of the sequence. So after blasting the feature ID, you are getting this answer. Okay, these are the features and these are the sequences of the features, right? First thing is that in the first table we saw that which feature ID was represented how many times in the sample, and here which feature ID, what is the sequence of the feature. Okay, so this is the thing. The sequence count is how much minimum length is maximum length in range. So these are the things that you can check in this in your file. Okay. Now we have what we have done. We have done the feature. We have made a feature table and we have summarized our feature. Now we will directly go to the taxonomic analysis part. So now in the taxonomy analysis part, what we will see is that which feature belongs to the which line. Okay. So this is the taxonomy analysis part. And for that we will require for analyzing, that means we will require a reference database. Reference database, that means the database is known. What it will do is that it will compare our field with the reference database. Okay. So for that we will be requiring a classifier, that is Vinci classifier. And this classification is on AI, that is it is pre-trained classifier. That means it has been trained by AI and it is called naive-Bayes classifier. And we will be using GreenLens. Some of them nowadays it has been Sylvia database also they are using. Okay. So So now what we will do is that first of all we have to download our reference database from reference classifier from the internet. What we will do, we will just type a duplicate and I will just copy the command and I will just download. Okay. So let it download. So it has been downloaded. The next part what we will do is that we will just do our taxonomy classification. So now there are two things. See, the first step is the process to assign taxonomy to the sequence in the feature data. There are two steps. First step is that we have to assign the taxonomy. Suppose this our feature data contains this taxonomy. Taxonomy means which are the bacterial present. So and the next what we will do is that we will train it. We will see whether our classifier is whether our taxonomy is like whether what we have done is uh, belongs to the database or not. Okay. So this is the first step. The first step is the process to assign taxonomy to the sequence of the guide to artifact. Artifact means the files. Okay, just remember guide to artifacts are the files. For that what we will use? We will use a name-based classifier and this is the plugin. Plugin means what that we have that are externally used. Okay, so these are the plugins. So what we will do is that I will just copy this code and okay. So now next what we will do after converting or getting our taxonomy file, we will see the interactive bar plot. Okay. So what is a bar plot? It will help us to see which taxonomy is present in which sample, like which maxims are present in which sample. So we got the two files. One is taxonomy.kxmcg, we got it. Okay. Now let me visualize it and let us see. Taxonomy. See. So at last we got our taxonomy. This is the feature ID that we got from our earlier section. This is the feature ID. And this is the taxonomy. That is, let us start from here. What we get? We get this one. Alpha proteobacteria. Okay. So what is the confidence level? This is the 0.00. This is the good confidence level. So this is the taxonomy. Now what we cannot understand from it, right? Still there is a problem of understanding. So what we will do, we will just convert this file into an interactive plot so that it makes us better to understand. Okay. So for that reason, what we will do, we will just convert it into a bar plot. Our input table will be this file and our output table will be visualization taxa bar plot of user. Okay. Okay. So we got this taxa bar plot of QZP file. Now let us take this. Okay. Now this is what still we are. It's becoming a problem to, a little bit to understand, right? For us. Now what we will do is that we will start with the bar plot and we will see an interactive. These are the sample name, and these are what? This is the taxonomic level one. That means now just we go to taxonomic level 4. Species, this one phylum, you are getting a phylum of Bacteroides. Then you are getting a phylum of Firmicutes. Suppose you want to check only the difference between Bacteroides and Firmicutes, what you will do? You will just click on this Bacteroides and Firmicutes and you check. Okay, here the first sample only it will be, but Firmicutes is there. Here also Firmicutes is there. Here also Firmicutes is there. But here in the last sample, Firmicutes will become less and Bacteroides will become more. Suppose you want to check, I'm just clicking see. You want to check the Proteobacteria. Proteobacteria is very less here. Very less in the second sample. Here it is more. It is little less. Here it is more. See the range. Just see the range. Here. It is like 55, 61 to here up to this. Okay. But here is more. Proteobacteria and Bacteroides, if you check. Here Proteobacteria is more, Bacteroides is less. But here no Proteobacteria. Now, as a researcher, once you get this data, you have to understand when you are writing a paper or something, you have to write, you have to see. This is the analysis thing you call it, right? Now you have to analyze why it is more and why it is less. Okay. Now we are coming up to the analysis part. Okay. So now again you can check it like this. See. Suppose I want to check this one, Bacteroides, and I want to check Proteobacteria. See, this is a Proteobacteria and I am checking a Bacteroides. Very less, right? So this is how we do. Okay. Now you can also increase the bar like this. Now let us see. This is the bar. Okay. And suppose this multiplier. So this is the bar here, right? So this is all about the taxonomy part. Now, the last thing what we will do is that we will check whether in the gut, suppose we have seen the taxonomic thing in the overall sample. Now we will see that whether there is a difference in the composition. Suppose in the gut we have taken, earlier we have in the sample metadata we have seen, that we have taken sample from the gut, from the palm. Okay. Now we will check whether subject 1 and subject 2, whether there is a difference in the bacterial composition in the gut or not. So this is called a differential abundance. That is differential abundance. Whether the bacterial abundance, whether there is a difference in the bacterial abundance of the two subjects in the gut. Okay. This type of question you have to do when you are doing the research. That means you have to check whether there is a difference between the two subjects or not. Okay. So for that reason what we will do, we will just copy it for that, and first we will create a table. That means a simple table, Q-sorted. That means the difference will be, first the difference will be calculated and it will be written in a table. Okay. And next the table will be converted into a visualization. Okay, so now we got the table, and next what we will do is that we will convert the table into a visualization. Okay, so this is the command. This is the command for converting, for seeing the differentiation part. So this is the command, time commission and com C, and here also next we will convert it to the part. First what we will do, we will convert it, the difference into a table. We will, from the feature table, we have, what we are doing is that we are filtering the gut table first. This is the first part. From the feature table, we are filtering the gut table. So this is gut table.qz. Okay. Next, what we are checking the differences in the gut table. How we are checking? We are checking the kind, composition, and com-C. We are checking the differences. And the output is in ANCOM subject.qz. That is the output. Now next we are converting the output file into our visualization file. This is the three step. First step is that you have the feature table, and from the feature table I want to see whether there is a difference in the gut microbial composition of the two samples. Now first thing is that from the feature table I am extracting the gut feature feature table. Okay, that part. Second thing, I have already extracted and I am writing written here gut table dot usage. Now the next thing what I will do is that I will see the composition. Now composition difference. For that what we will do is that we will use ANCOM dots and we will check the this part and we will see the composition part whether there is a difference or not. Okay. Once we have used this ANCOM.c and we have already calculated the difference in the gut, our table, our this one analysis result will be saved as subject.qza. And next to visualize it, we will convert the subject.qza into a bar. Okay, so this is the thing. It will take some time. Yeah. So we got the ANCOM subject.qz. Now what we will do is that we will convert it to QZB file. Okay. So now we got the bar plot subject.qzb file. Now let us check it and let us see. So let's see. So these samples are enriched. Okay, these are the sample IDs that are enriched, and these are the sample IDs that are depleted. That means these are the subjects, subject 1 and subject 2. These are the sample IDs that are enriched and these are the sample IDs that are depleted. Okay. So these are the feature ID. Feature ID. And next what you have to do, you have to see the feature ID. You have to go to the back table, taxonomic table, where your feature ID has been converted to taxonomy, and then you have to see which belongs to where. Okay. Another thing is that what you can do is that you can convert this feature ID to the taxonomic table. Now let us do. We are converting this feature ID into taxonomic table. Up to level 6. See here, it is a table. We are converting that table.qz, we are converting it to taxonomy.qz. We are inputting the taxonomy.qz also, we are inputting the table.qz up to level 6, and we are collapsing both the tables. So that, see, what I told you is that you have to go back to the feature taxonomy table and you have to check which feature table, which feature represents which taxonomy. So without going there also, what you can do is that you can directly type it and you can. Yeah. So we got the first part. Let's we convert it into the future. First we merged our taxonomy table with our feature table and then we will convert it into a visualization table. So we got it as 16DA bar plot subject of future. So this is a visualization table. And yeah. So remember this table, feature ID subject. Okay. Still now we don't know the taxonomy, which feature ID belongs to which. Now we will check it, which feature ID belongs to which. Yes. Yeah. Now we got it. See. These are the enriched, right? These are the enriched. That is Humidicus, Bacteroides, Bacteriocyte. So these are the enriched. And these are the depleted. That means this thing, these phylums are not found in the both the samples. Okay, this phylum has an abundance in both the samples. This phylum. But this phylum is less abundant in the sample with respect to the gut microbe. So this answer is what? So what now? Now the next question will be what is making this phylum, this bacterial abundant and what is making this bacterial depleted? This will be our next question and for that also we have to do some experiments. Okay. So now we have completed this moving pictures tutorial. Okay. So okay. So we have completed this moving pictures tutorial. So yeah. So if you have any question regarding this tutorial, you always
There are any queries? Anyone can ask in the chat box.
Session is now open. Type in the chat box or they can simply unmute. You can unmute yourself. You can ask queries if you have.
I guess there will be doubts regarding this. Once you don't means once you practice, everything will be clear. Most of things are new, I think.
I hope participants are still on the other end. If any one of you can please unmute yourself and say a few words. Otherwise, if there are no queries, then I can implement the session. And then Zachariah, please continue.
Thank you, ma'am. And thank you, sir, for your valuable time and insightful session. And
Thank you.
now I request Ms. Malika Barua to end the meeting with a formal vote of thanks to Ms. Malika Barua.
Thank you, Zakaria. Good afternoon, everyone present here. On behalf of the I extend my gratitude to all who have contributed to today's project in meaningful and successful. I would like to extend my gratitude to our distinguished research person, Dr. Anisha for taking out valuable time from your busy schedule and sharing your insightful knowledge with us. Your words have truly enriched our understanding.
My sincere thanks to our coordinator for their cooperation, cooperation and supervision in smooth conduct of this project. I also acknowledge the valuable assistance of our faculty members who have worked behind the scenes and ensure that the arrangements were made efficiently. Lastly, I thank everyone present here for being a part of this program and for your patient listening. Thank you, everyone.
Thank you. Thank you so much, sir.